Thank you, Mona. Good afternoon, everyone. Have you ever wondered how do scientists test drugs and treatments for life-threatening diseases like cancer? I mean, they're experimental, so of course you can test them inside the lab on a couple of cells or maybe even a plate of cells. But how do you know for sure that they will be safe for administering to actual patients who have complicated organs and functions? So my talk today is about a liver-on-a-chip platform, which I developed during the course of my master's project at ETH Zurich, which uses a novel fluid handling technique called microfluidics to enable liver samples from patients to be cultured for long-term drug testing studies outside of the body. So here I'll briefly go over the outline of my presentation. I'll talk a little bit about the liver's importance in the human body and how the cancers affects that and the prevalence of liver cancer in the society today, as well as some existing tumor models that are used to test anti-cancer drugs for the liver, and then the design of the microfluidic platform that I worked on during the course of my project, and then an outline of a culture experiment which I conducted, and the results of that one at the end. So talking about the liver is a main important organ in the human body. It's in fact the largest solid organ, and it forms a range of important functions, as you can see here. So the first one is a production of a protein called albumin, which is an important component of the blood. And the liver also creates another a protein called bile, which is important in the digestion of fats in the digestive tract in your body. The liver also plays an important role in the metabolism of drugs and toxins, so it kind of pacifies the drugs and toxins and to get rid of their activity once they've completed their activity in the case of drugs. And it also plays a key role in the regulation of blood glucose, and it also stores several vitamins and minerals as well. So when you consider cancers of the liver, they're fairly common, similar to other organs. And here we can see the statistics. Approximately 900,000 new cases were diagnosed in 2020 alone, worldwide. And this is the sixth most common type of cancer worldwide. And additionally, on top of that, 800,000 deaths were caused annually as a result of cancers related to the liver. And so this, from these two statistics, you can see there's an extremely low patient survival rate of liver cancers. And this highlights the need for developing better treatments to combat liver cancers. And it also poses a substantial healthcare burden, with 4 billion euros being associated with liver cancers in Europe. So when it comes to models to be able to test anti-cancer drugs, these are three of the most common primary tumor models which are used. And by primary, we mean the original sample for testing is acquired from an actual patient with cancer. So in the case of patient-derived xenograft, which is a type of animal model, this sample is implanted into an animal host, and then you expose the drugs to the animal. And the main benefit of this is the fact that you can test the drugs on a systemic level on the whole organism. But the disadvantages, of course, is the fact that the immune response is different between the animal host and the human. There's also a lot of regulatory issues, and it's also a very expensive method. And a second approach is to use patient-derived organoids, which is a high-throughput approach, because what you do in order to make these patient-derived organoids is you take the sample and you dismantle them to separate the different cell types and recreate them into a three-dimensional structure like this. And it allows you to produce a lot of these PDOs from a very small amount of patient material, so you can test a lot of drugs at the same time. But then again, you lose the original tumor interactions because you kind of destroy the original sample to separate the cell types to make a structure like this. The model that I worked on is called precision cut liver slices, which is where you basically slice this sample into thin sections, so that preserves the original architecture of the cells in their native form. And that's the main benefit of this. But these PCLS are very sensitive to a lot of environmental conditions like temperature, pH, etc. And so that brings us to my presentation. The aim of my presentation, which was to develop a microfluidic flow culture platform, which allows us to maintain these PCLS in a robust environment that you can actually test drugs on them. And this is the platform that I developed, which consists of 4 parts. The medium reservoir, which basically holds the culture medium for providing the nutrients to the cells, and a pump that pumps this medium around in the loop, and an oxygenator, which provides the gas exchange, and the culture chip that actually holds the slices. So the main benefit of this type of system is that there's a continuous exchange of nutrients and oxygen into the culture chip, which has the cells. And it's also done in a very small volume, which I'll show you in the next slide. And this replicates the physiological conditions that the cells will experience in the body much better than a static culture chip. So here we can see the design of the culture chip. So it consists of a plastic chip like this, which contains 3 wells that can hold 3 precision cut liver slices upside down in these hanging drop form. And then the medium flows from an inlet to an outlet. So that's how the medium gets to the slices. And they can be arranged in a parallel assembly like this for higher throughput. And here we can see a picture of the overall setup that I mentioned, that I showed you in the schematic, with the peristaltic pump, the oxygenator, and the reservoirs, and the culture chip. And you can see here the tubings that are used to connect these are very small. And this allows precise control of microliter volumes, and hence the name microfluidics. And now we come to the actual culture timeline. So in the experiment that I conducted, we received, for example, notice of an upcoming surgery 3 days before, and that's when we start to fabricate the new chips, sterilize them, and set up the experiment. And then on the day of the surgery, the tissue goes directly to our collaborator, who sliced the samples into these thin sections. And then after that point, they were transferred to our lab, at which point we embedded the slices in our culture chip and then put them into the microfluidic system inside this box. And then we cultured them for 50 hours and sent the slices back to the collaborators for analysis. And now I'll show you some pictures of the analysis. Here we go. Yeah, so these are 2 representative results from the present trial, which was done for 48 hours, and a previous trial for 72 hours. So in the present trial, we can see a lot of healthy liver cells, which are characterized by this darker color of the pink, and also the presence of nuclei. Whereas these dead cells don't really have that nuclear stain. You can see the dark purple is not there. So based on these proportions of healthy cells and the dead cells, it's possible to characterize the percentage viability of each slice. And that's how we report a viability table like this. So in this table, we can see out of the 5 samples that were analyzed, 3 of the samples showed very high viability and 2 very low ones. And we consider this a very positive result, considering the number of samples that we have here, because these 2 can be considered outliers, and these likely attributed to the handling issues, because this is a very complicated system. The loading and unloading can have effects. And so that brings us to kind of the future work that can be done. And these are some further improvements to the system that needs to be done in order to improve the system further, by increasing the culture period for up to 5 days, for example, to enable longer tests, and to test with actual liver cancer cells, because the samples that I used were healthy ones, and to also optimize the analysis protocol to avoid outliers such as the ones I showed you. When it comes to the future outlook, I think this research shows proof of concept that this kind of flow perfusion culture enables better maintenance of the PCLS in their healthier state. And it also enables low-cost and animal-free platform for testing drugs in a more humanized fashion. And it also enables more clinically, in that sense, enables more clinically relevant drug testing studies. And that concludes my presentation. I would like to thank my supervisors and my collaborators for enabling this presentation. Thank you.